Hey everyone, welcome to Lab 8. So I just wanted to go over a few things to make sure that you guys have everything that you need to do this lab. I will try to post some videos in addition to the lecture videos that you should have watched, but I would recommend watching the videos. Hopefully they'll be pretty helpful for you guys. Anything with really anything in this unit, animations and videos are probably really going to be helpful, but especially for mitosis because you're looking at a whole dance of cell division, mitosis is really helpful on video. So you guys are going to download this from the assignments or under resources, and you're going to go through it. It does involve quite a bit of drawing, so you're welcome to do that either on a piece of paper, and you can take a photo and upload that as part of your assignment, or you can do it in Paint, or you can do it however you want to do it. On my computer, I have a newer version of Word. I'm not sure what version this is or if you do or don't have it, but if you see right here, there's an option to hit draw. Let's see here. Draw on Word. And that's probably one of the easiest ways to do this. Okay, so you can select like a pen. You could even just insert like text boxes and arrows, however you want to do it. You can insert a text box here, and then you literally just, like, let's say, oh no. Let's say we want to put G subscript 1, okay, and then I want to move my text box up here. I want to let me grab it. There we go. And I can move it to wherever I need it to be, right? So you can do something like that, or you can hit this draw. I think the draw is really easy. I can draw with touch because I have a touch screen. If you don't have a touch screen, then you can do something else. But basically, the first part is asking you to color and label. Let's see here. Yeah. So you're going to color and label here. The coloring is optional, so you could do something like color this red, right? However you want to do it, where I can still kind of read it, and then color G1 red. So just as an example, you guys know what this is. And you can print this out and do it. You can draw on your own piece of paper. You can use some colored pencils or markers. Get creative. It doesn't have to be so creative to get the points. It just has to be correct to get the points. But if you're coloring and doing stuff like that, this usually ends up being a really nice study guide or study tool for your exams as well. So I would put in a little bit of time to it if you want to save some time down the line when you're doing class activities and the exams and quizzes. So you're going to label it just like that. The other thing that you need to label on this first part are the checkpoints. Okay, so there's 3 different checkpoints that you should be labeling that are in the cell cycle. The first one, as a hint, is happening right here. So I'm just going to say CH for checkpoint. Checkpoint 1, and then tell me what that checkpoint does. Okay, there's 2 other checkpoints on here, so make sure you label them all appropriately. Again, you can label this however you would like. Okay, then we're going down here. This is just matching up the phases with the description that's happening on the right. So pretty straightforward. It might take you some time to go through it, but again, this will be a helpful study tool. And this is just going through all of the different phases. I would probably recommend doing the first page first, but you'll see that some of these things are defined here. So as long as you're getting them correct, that might also help you with the first page. Okay, it's going through and defining every part of the cell division cycle. Okay, and then number 3 and number 4 are simple questions based off of that. And then we're gonna get, oops, I'm still drawing. Okay, let's go back to select. So I stopped drawing on this page. Okay, then we're getting into part 2. So this is where we're going to be observing mitosis in onion tip cells. So since we're not in the lab, what you're going to do is you're going to control click to follow it to this page. Sorry, Max, you might do something different. It should look just like this. If this doesn't work, you might have to clear your cookies, restart your browser. You might have to look at it on a cell phone. Do what you got to do. You need to get to this page. Okay, and the link does work. I've tried it multiple times. I've had other people try it, so it does work. And then you're going to go here, follow the directions. Basically, you're going to click here to look at the root tip. So this is the root tip of an onion cell. We're going to click on it one more time to magnify it, and now we can see all of the different cells that are in here. And if you were in lab, this is exactly what you would be looking at under the microscope as well. So you can see these cells are in different stages of cell division. So you're basically going to go over the cells until you have one that has that little icon, the little click icon. You're going to click on it, and then you're going to tell me what stage of cell division this is in. Interphase, prophase, metaphase, anaphase, telophase, / cytokinesis. So here, telophase and cytokinesis aren't really differentiated, and to tell you the truth, when you're looking under an actual microscope, it's really hard to tell. So you're going to say which one it is. Let's go ahead and say it's metaphase. It is not. But the nice thing is it's going to tell you it's not metaphase, and then it's going to say what happens during metaphase. So now we can click it again. We'll click it again, and now we can say prophase, right? And it just tells you correct, this is a prophase cell. Okay, so this is kind of a good way for you to study what these actual cells look like. You might see something similar to this on the exam where you're seeing more of a real cell versus an animated cell, and they are kind of hard to tell the difference, right? So I would practice this. You can see different cells. I don't know where all of them are, but most of them are the more well-defined kind of bigger cells like that your eyes are probably naturally drawn to anyways. So go ahead and click on those, go through this activity. And then once you're done with that, you can come back to here, and then you're going to be sketching out what those cells look like in each of those phases. So you can basically just look at it here. So say we picked this one for prophase, right? There's our prophase cell. So we can basically just draw that into here, okay? And again, digitally draw it on Paint, draw it on Word, draw it on whatever program you have. I know a lot of people can also draw over PDFs, or you can draw it by hand, but you will be drawing some in this lab, okay? Then ask you a question here, and then we get to, we're gonna be doing some calculations, okay? So we're looking at cell division in cancer cells. As you know, cancer cells uncontrollably divide, and you'll get a little bit more information about all of that in the lecture. But you'll go through, answer some questions, but you're actually going to have to do some calculations once you get to this part here. So the data is all really collected for you, but you have to calculate out the number of minutes and the % of the cell cycle that each stage of mitosis accounts for. So you're going to look at healthy chicken stomach cells versus cancerous chicken stomach cells. Okay, so the way you're going to do this, the instructions for it are right up here. This is how you are going to calculate out these numbers that are down here. Okay, and they can be exact. You can round them to the nearest tenth or 100th. I don't really care as long as I can understand that your answer is correct. And make sure that all of your answers for this add up to this and this. Your percentages should add up to 100% on both of these, and your time should add up to the total time. So for interphase, as an example, we're looking at 440 cells, right? So 440 of the cells that we see in this example out of 500 total cells, okay? And normally in lab, you actually go through and you count cells in the onion root tip cell. I have taken that out of this lab for you so that you guys don't kill me. So you guys can be really happy that you only have to do this here, which is already giving you the data. So 440 divided by 500. If you divide that, you get the % of the cell cycle. So 440 divided by 500 times 100. Sorry, my 100 is going to disappear because it's out here. But for that, you should get, I believe it's 88%. Yeah. So here we're going to have 88%. And again, you can draw this in here, you can type it, I don't really care either way. But you might want to check with each other to make sure that you've got the math down correctly. So 88%, and now what we want to do to find the total time in minutes is we're going to do 440 times 500, or we can take that 88%, which will be 0.88, and multiply that by 625, which is our total time down here. And once we do that, we should get the percentage of time that our cells are in interphase. And that should be, I think it's 550. Yeah, 550. Sorry, I know that's hard to read. So there's an example. Your first one's done for you, an automatic 2 points. So you just need to figure that out correctly. So you can see that most of the time is spent in interphase, which we know already, right? The interphase takes the longest amount of time because we're going through G1, S, and G2. Okay, and there's a couple checkpoints in there as well. So you're going to go through, answer those, or calculate out those, and then you're going to answer these questions based on what you just did. The last question, keep in mind, you're looking at cancerous cells versus, well, all of these questions, you're looking at cancerous versus normal cells. But think about what cancerous cells are doing, right? And what they actually are. Cancer cells are abnormal, and they're really, really fast growing. They're dividing really fast. They're dividing really uncontrollably. So think about the kinds of things that cancer cells might be skipping, right? They're not skipping a lot of the cell cycle because they're obviously going through it really quickly. But there's something really important that they're skipping that you talked about on the very first page that you marked that allows them to keep going through the cell cycle instead of stopping themselves. Okay, so there's some hints for that question. So overall, that's the whole lab. I know that this stuff can get a little bit confusing, but this lab will be a really good study guide for you, especially this first and second page and that resource online as well. So if you have any questions, please let me know. You can always stop by office hours or you can email me. I highly recommend doing this with a group. You guys can organize a Zoom call and do it together for an hour, or you guys can do it together or do it separately and consult with each other, however you guys would like to do it. I do recommend making sure that you guys have correct answers for this because it will be really helpful for you in the upcoming weeks.